Hello, everyone. Welcome to Jack's Career Chats, a series where we highlight the diversity of jobs, skills, and opportunities in the world of biomedical research. Today, I'm talking with Dr. Kevin Anderson, a postdoctoral researcher working in the lab of Dr. Roel Verhoek at the Jackson Laboratories campus in Farmington, Connecticut. Kevin studies brain tumors, specifically gliomas. He has a background in biomedical engineering and genetics and combines his skills in biomechanical and genomic approaches to analyzing brain tumor heterogeneity and disease evolution. Kevin, thank you for being here. Welcome.
Yeah, thank you.
So let's talk by this exciting work that you do as a scientist. You research cancer, but specifically gliomas, which are a certain type of brain tumor. Tell us a little bit more about this special type of brain tumor that you study.
Yeah, sure. Well, it's one of the more malignant of the primary brain tumors. Like a lot of times when you think about primary brain tumors, there's sort of two general categories. There's the kind that starts somewhere else in the body and then spread to the brain, and there's the kind that originate in the brain. And so of the kind that are originating in the brain, gliomas are one of the most deadly, unfortunately. And so part of the issue with that is something called heterogeneity that you mentioned in the title. And so that's what I focused on. And so what that means is if you look at the molecular background of a cancer, it's not just one particular background. There are different subsets of cells that have different backgrounds. And so what we're trying to understand is, are there consistent connections between those different subsets of cells that we can use to sort of improve on treatments? Because right now, the only real thing you can do is you can get surgery and you can remove most of the tumor, but the problem is that there are specific cells that they spread out further from what you could see on, say, like an MRI. And so you might have some tumor cells that exist after surgery, or you give them chemotherapy and you kill most of the cancer cells, but some remain because they have different molecular backgrounds. And so even with the current treatments, the lifespan after diagnosis is only around like 15 months or so. And so we've been trying to look for ways to improve that.
And so what's the difference in the molecular background that you mean? Do you mean the genetic code of the cells that you're looking at?
So you can look at it in terms of genetic mutations and then also changes in sort of the regulatory structures. And so that's, for instance, DNA methylation, which are tags that are present on whenever there's the letter C in front of the letter G, or which call that a CpG dinucleotide, or CpG for short. When you have these markers on these Cs, that it can change how these codes, the genetic codes, are written out. Like, for example, in the promoter regions, if you have a lot of these markers, it can say, well, this particular gene here, these markers can signify you don't write out the instructions for this particular gene. So even though the genetic code is there may be changes in the regulatory system. So the code looks one way, but something different is happening. And so you can just get these different molecular backgrounds.
Yeah, I see. So that's epigenetics, right?
Yes, yes.
So that can change even the output from transcription or from translation, what you get from the cell, without changing the actual genetic code itself.
Right, right. So it's definitely a tricky issue. And so you can look at it from different ways. And so our lab focuses in on looking at changes in those genetic codes, as well as the change in the regulatory mechanisms.
And so how do you study? Are you doing sequencing or imaging, or what are the types of techniques that you're doing to get a glimpse at these cancer cells?
We primarily do DNA sequencing, and so, or DNA and RNA sequencing, but sequencing. Yeah, so we partner with different hospitals and other institutions. Like, for instance, we have different collaborations where we'll work with surgeons who see these patients, and when they get the tumor removed, they'll obviously take a section of this tumor because you need that for the doctors to do diagnosis and test for, which treatments might be best. But then they'll also send us remaining parts of this tissue. And so we can use that to sequence, and then we can use that for testing different things or for looking at patterns that we might see amongst these different patients.
Oh, cool. So you're looking at the actual patient tumor samples themselves.
Mm-hmm. Yeah. And so
do you do any sort of like mouse model or animal models, or is it all like clinical human samples?
So one thing that you can do for different tests, because obviously it can be hard to keep track of when you'll get samples from people or how often they'll come. And so one thing that we can do is that we can take some of these cells from these tissues and then grow cultures from them. So you take a few cells and then you can grow what we call glioma sphere cultures, where you just take groups of these cells and you grow them up in this controlled condition. And then you can freeze them down, you can get some later. And so sometimes with partnerships, as opposed to getting the specific tissue, we have these cell cultures that we can use.
Oh, cool. Do you
Yes,
work on that as well? Do you work in this on the cell culture side?
I don't physically do cell culture stuff, but I have worked with, so some of our lab works primarily on computers. Like I'm almost entirely on a computer, but I will walk into a wet lab to help out with something. And so I've been working a little more with actually seeing it physically instead of just doing everything on the computer. And so we like the one cool thing about JAX is that they have all these different departments that do stuff. And so I've been working with the single cell biology lab because we'll do some experiments. And so one project in particular, there's a group of surgeons in Luxembourg, and they shipped the tissues. So I have to track the FedEx and make sure everything's still frozen nicely. And then I'll work with some other people in my lab or other departments to hand them the cells, and then we can work on the experiments we're doing. And so I'm sitting down and watching it, but still not really doing anything physically. But
Yeah,
yeah,
but you still have to know what's going on, and it sounds like a really nice opportunity for collaboration with different types of scientists.
yeah, it's pretty nice. And just being able to sit there, because sometimes you'll hear about procedures or you'll see stuff where you're like, for instance, what I've been working on is nuclei isolation lately. So you take the nucleus of the cell specifically because you want to sequence that for certain different technologies. And so I got to sit down and actually watch as they walk through it and went through the different steps to do nuclei isolation, so that when I see something about a protocol, I can sort of know how that could affect the sequencing. So it's pretty cool to see.
Yeah, awesome. So tell us a little bit then about what you do. You said you're more on the computational side.
Yes, almost entirely. And so usually once we partner with either people in our group or the single cell biology group or other like GT, genome technologies, we work with people to actually sequence the tissue. So someone has to go through and run through the different steps where you add different reagents and do the different chemical reactions you need to either get the DNA or the RNA or whatever you're looking for. And then they'll run it through the sequencer. And so I'll start at the step where we get an email from someone that the sequencing is complete. And so what you end up getting is these files. Like for instance, if you're looking at genetic mutations, it's these really, really large text files with just the G, C, T's, the different letters of them. And so those are, and so I'll take those files and then I run them through different softwares. So I write and adapt various available software programs to try to analyze these sequences. And so the first thing is to try to condense all that information down to something that you can actually interpret or get some understanding from, because if you just look at the text, it can be a bit overwhelming just to see all those letters. And so just trying to write something to get it in the various programs so that we can analyze these patterns more specifically. So that's where I come in.
And did you have coding experience before you came to this position? Like how did you learn how to do what you're doing now?
Yeah, so I started as a biomedical engineer in undergrad. I liked biology and I knew I liked working on computers, but there was a bunch of stuff that I liked and I kind of didn't know what I wanted to do more. And biomedical engineering kind of felt like taking an intro to everything. And so I got to take chemistry classes, more mechanics focused classes. Like it was pretty across the board. And one of the classes I took was mathematical modeling of biological systems. And so we worked with MATLAB specifically, which is one particular type of software program. And it was, for that class, there was a lot of examples on, say, like analyzing cardiac function. So like if you could actually take the signals of heartbeats and try to look for patterns that you could say, figure out when the rhythm was off or stuff like that. And I thought it was pretty cool. And so I didn't really know about a firm way that you could use computers or coding and stuff like that. And so I started taking a few more classes like that. Like I took one on vision, like signal processing, like trying to code or adjust how like I guess I'm trying to, it was kind of a few different things, but I guess the main thing is sort of if you look at the brain as a computer and try to run codes to replicate some of the things you could see. So like one of the hardest things was a project on trying to take static images and put them together in like a 3D form, like the way you do with binocular vision. And I thought it was really cool. And then I ended up joining a lab that worked in molecular dynamics, which is looking at the behavior of proteins that are really, really, really small scale and over a small period of time. And so you're running code. And what I was looking at was how proteins can enter a cell membrane. And so you actually model that so you can see the proteins folding when they approach the membrane. And this is over like nanoseconds. And it was just interesting how long and how much computing power was needed to look at that sort of thing. But I thought it was really cool. So that kind of took me into a path of, I guess, trying things that had vaguely similar themes of looking at biological mechanisms from a computational approach. And then I kind of sort of fell into where I am now.
Yeah, that's cool. So as someone that's always been more on the biology side, I'm always fascinated because I feel like I learn more and more about the integration with the, engineering or the computer science or all these other elements that I always thought were so separate, but they really can be integrated and then can be so useful in biology and open, more doors to research and careers. So it's really fascinating to hear that, protein modeling, for example, that you talked about.
Oh yeah, for sure. It was definitely neat because you meet people from both ways. There are some people that were biologists fully, and then they started doing coding stuff, and there was other people that are just completely computational, and they look at it purely from a numbers perspective. And there was definitely something that I could learn from all of them. And so I'm glad I got to try the different, like either working in labs or doing rotations or just sort of just kind of getting a taste of what work in that area was like. It was pretty cool.
Yeah, so it sounds like your path in biomedical engineering was like a really good segue to kind of show you, like you said, the breadth of different things in STEM, basically.
Yeah, I feel like different people I met looked at it different ways. There are some people that sort of they were sort of drawn to a particular technique, and then eventually they find a research question. And then there's others that were really stuck on this is the biological thing, or this is the disease, or something that I care about, and then they sort of discovered new techniques. And I feel like I was kind of more in the former thing. I was just kind of I was really into coding. I remember one day I was like taking notes in class and something came up about careers or something like that. And it was just a simple thing like me trying to take notes or think about how you keep up with all the papers. And my professor was like, Kevin, you're always carrying your computer on you. Why don't you just do it on the computer? Because I was just trying to handwrite. And that's when I was like, oh yeah, duh. And it was kind of an interesting little thing. And it sort of led to this conversation of what's the stuff that you like to do already, and I always was taking around a computer. I played video games a bunch, and I would actually get into the hardware of my computer, like change a new graphics card or put a new like just the little tweaks and stuff like that. And so, you pick up very, very, very minor things in coding. And I took a class where I was like, oh no, I really like this. Oh, what's the stuff you can do with it? And then that's where it was like, oh, molecular dynamics, and that was one thing. And then when it came, I did that for the final few years of my undergrad. And then that problem was a more specific one. And I guess I was kind of looking to see what else I could work in that was similar. And so when I was applying to graduate schools, you sort of see the programs and stuff that people are working on. And so one of the labs that I applied to was doing fluid mechanics. So using computational approaches to model the flow of cells and blood vessels. And that seemed pretty interesting too. And so, I worked on that, and that's how I got into working on cancer applications. So I kind of went techniques first and sort of fell into a biological problem that was pretty complex and interesting.
Yeah, it's great to hear a path. And what I really liked about what you said too before about your skills and just thinking about what you enjoyed in your life and how you did things, like the video games, and then just how much you liked that and how you would kind of be hands-on with, changing out your graphics card and stuff. And that kind of helped shape you a little bit into the path of coding and being more hands-on with the computational work and, just being observant about taking notes on the computer, those types of things. Like they all can kind of help you realize like, what you're good at or where your skills lie and direction that you could go into.
Yeah, I thought that was pretty good advice. Just sitting down and thinking like, okay, so this person and someone else also mentioned something like that. Like if you can put like the pros and cons on the stuff that you do all the time, or like what you like
Yeah.
about what you're working on. And yeah, it's like a hard puzzle, and I like doing those. Strategy games, board games, just regular puzzles, and I can look at coding sort of like that. It's very it's very structured, and there's a lot of leeway, there's a lot of creativity, and there's a lot of ways that you can approach this sort of thing. And then other people obviously feel the same way because there's a bunch of software out there.
Yeah, absolutely. And that's like a good a common trait, I think, in science is that discovery, that creativity, the problem solving. Those are things, because you're doing experiments, you're discovering something new. And those are the feelings that a lot of scientists get. So they're all often drawn to that sort of feeling.
Definitely good opportunities for problem solving.
Yeah, yeah. So you have a couple of slides to share about your research. Do you want to go back to that and tell us a little bit about those the cancer cells that you study, the tumors that you study?
Yeah, sure. There we go. So, like I mentioned before, is the big issue with looking at glioma is the thing heterogeneity, how there's differences between one particular set of cells or another in a given tumor. And so this is part of one project that I was working on where we partner with surgeons that take MRIs. And so MRIs is where you get this imaging feature where you can have the signal where you have a more light that's shined on a particular image for what you're looking for. So here, there is a certain imaging modality that makes the tumor brighter. Now, this only works for the main core of the tumor, and so obviously there's going to be some tumor that's not lit on this MRI image, and so that's one of the things is where you can capture this mostly. And so when surgeons are preparing to do a surgery, they're using these MRIs to dictate where they're going to remove the tumor from during the particular surgery. And so this left was an example from one snapshot of looking at an MRI of a particular brain. And so the tumor volume, I don't know if is my cursor? I don't know if you can see that or
Yep,
not.
yep.
But the cursor is here showing where the tumor is highlighted. And you can also see a part here that's part of this main tumor that's separated. And so even on this scale, you can kind of see how it's not always in one place. And so it's
So
pretty,
is that called metastasis, or why is
this
it separate like that in space?
So this part right here, oh, sorry about that. This part right here, I wouldn't consider metastasis. This is just part of, so these type of gliomas, the ones that are more aggressive. So gliomas they can be graded within 4 grades. And these grades 2 through 4 are considered diffuse. And part of that is because they spread out. And that's kind of just a natural thing that occurs with this progression, is that these cells just continually spread out. And so you'll just see this. Metastasis, I would consider more of a shift to a distant organ or region. And so I wouldn't necessarily call this that, but it is somewhat similar in that you have these cells that are moving along, say, blood vessels or other vasculature or lymphatic networks to get to different locations.
Okay.
And so one of the issues that the surgeons were having, and that part of the study was founded on, is that depending on where you take the biopsy, you may get a different diagnosis for the subtype. So it'll register as a glioma, but maybe it'll register as a different type of glioma, and that can affect which treatments they think will or won't work. And so what we're working with them is to see if there's some consistency. And I guess it's a few questions. So one thing is taking one biopsy enough to get a firm grasp of what the molecular background of the whole tumor is? And unfortunately, the answer seems to be no. And then the second question is, well, how different are these different tumors from one location to the other, even within the same particular tumor? So if you took a biopsy here versus you take a biopsy here, is the molecular background the same? And would they respond to treatment the same?
Mm-hmm.
And so what these surgeons did is they took multiple biopsies within a given person along different areas. So like you might start in the center and you'll move further and further away from the tumor. And at what point does this molecular background seem different? And so they gave us the actual images they took from the biopsy. And then we also did sequencing from the biopsies that they sent out. And so what this first part I was doing is sort of visualizing where these biopsies were taken relative to the tumor, and then what the molecular background of that is. And so over here I worked in MATLAB to sort of render the brain and then render the tumor and then render the biopsies so that you can see physically where the distance is. And so what you see over here is this red is the tumor volume that is showing up from the MRI. So this part would be recognized as tumor by the surgeons that are looking on this after taking the images. And then these shapes here were the different biopsies. And so what I went through working is trying to have this sort of physical code. And so the shape is one particular way that you can classify where these tumors are. And so tumor core is the closest to the center of the tumor. And so tumor cores can be associated with something called hypoxia, where there's not a lot of oxygen. And so you'll have more necrosis or dead tissue. And this can be considered where the most aggressive cells of the tumor could be. And then further out, you see the triangles. This is on the periphery, so this is on the edge of the tumor. This could potentially be cells that are growing, that are trying to get this tumor to spread, or they could be cells that are sort of spreading away from the initial tumor. And so it's possible that the ones that are on the periphery that are spreading away have a different background that's allowing them to move further and to survive longer than the ones that are at the center. And then further out, you have outside of the tumor, which is just, we would consider it normal, but that's a nice, interesting question when you get into what makes it normal. Because these actual tumor cells, they send off signals to cells nearby them. And so even if it's quote unquote a normal cell, it may be receiving signals to say grow faster or resist treatment, or it may have changes. So even if it's a normal cell, it doesn't quite behave normally. And so then that gets into this broader issue of how does treatment affect it? Should this be removed when surgery is applied? And in the brain, that's obviously very tricky. You don't want to take too much away because that can have negative effects on the patient.
This
And
is
so,
super cool, Kevin. So you made this image on the right. So you took,
yes,
this is your like MATLAB coding rendering of the brain, and then the tumor, and then the sample biopsies that
yes.
they took.
And so it was a mixture of applying different stuff that exists in MATLAB for taking these type of images and making the volumes, and then adjustments that I made to say, like draw out the specific tumor volume, like trying to dissect from the code itself, how you separate this volume from the rest of the brain, and then how you do the different renderings. And then combining that information with what we get from sequencing in order to give the labels. And so we can kind of see that you see all these shapes are blue here. These would be given the same classification by methylation, those tags that I mentioned previously, whereas this green one would have a different classification by methylation.
And those classifications are coming from your sequence analysis, and you looked at all the biopsies, and you're just kind of comparing what's the same, what's different, how can we categorize them?
Right. So we're looking at the patterns of methylation to see what are different. And so, for instance, this GSIMP high to GSIMP low, it's called the glioma CPG island methylator phenotype. But that just means that in certain regions of the genome, you'll have increased methylation as a result of this disease progression. And so that background ends up being different. But this is not the genetic code itself. This is in the methylation.
Yeah, that's that epigenetic mark.
And
Yeah,
then,
this is super cool. I mean, I think, we think of tumors, and I know that, a liver tumor will be different than a brain tumor, but you kind of think of tumors as just one thing, and then they're tumor cells, and they're bad, and they're all the same. But this is showing us that even within one tumor in a small area, it can be very different, all the cells within.
yeah. And that can definitely be a problem when it comes to treating something, because you may give someone chemo or some specific drug that targets most of the cells, but those ones that escape that have that different molecular background, they're not going to be affected, and they're going to grow back more resistant overall. And so that's part of one of the main issues with treating people with cancer, particularly this type of brain cancer.
Yeah, that's a perfect segue into my question about treatment. So how do you use this information in order to kind of learn more about therapeutics and how to treat something like this?
So I think one of the general practices, because I mean, it's a lot of, it can be tricky to try to find the ideal treatment plan for someone. And so sometimes you'll have to go by the numbers. What's the most popular thing? But one of the things that stuff like this can do is allow us to narrow down, okay, so maybe the most popular treatment works 80% of the time. And so by a numbers game, it makes sense to try that first and see if it works. Whereas now, if we say, take a look at this, well, if this methylation type is here, there's one type of chemotherapy treatment called temozolomide that's used with people with this type of brain cancer. Well, you can look at certain methylation patterns and predict whether or not that treatment's bound to work. And so you can say, oh, well, if this particular region is methylated, this treatment may work better. Whereas if this particular region is not methylated, this treatment probably not works, and so maybe we should look towards an alternative. And so you can save time, you can increase the comfort of the patient. So that's just like a small case example where you can sort of make their lives better by looking through these molecular backgrounds first. And then ideally, you would want to get to the point where you can narrow down, okay, this set of treatments will work, would be the
Awesome.
ideal scenario.
Yeah,
And
that was really clear. That makes a lot of sense.
thanks. And then one other thing I just to mention is that the color of these borders is the classification based on RNA sequencing. And so that's, well, I should be specific, predicting the RNA sequencing based off the methylation patterns. And so this is estimating what the actual gene products that are being produced by that cell type are. And so sometimes the regulation can change what genes are expressed or how much, and then, sometimes not. And so we're trying to estimate both of them just to look for differences there as well.
I see. So those epigenetic marks, those methylations kind of control which genes are on and off, and that thereby controls which genes are expressed and what RNA is made. So you can predict based on those epigenetic signatures, the methylation marks, what
Yeah.
RNA you'll see or not see.
Yeah.
Cool.
And then, so this is one project that I've worked on a bit. And then just to talk about a different project. And so again, on this topic of heterogeneity. And so this is called a phylogeny tree. So whereas before, the last project I was looking at is talking about methylation or these regulatory mechanisms over the genetic code, this is looking more specifically at the genetic code itself. And so, like I said, sometimes you have changes that occur over all the cells, and sometimes you have changes over a subset of cells. And this is how we try to quantify which cells are being affected by which molecular changes. And so with this tumor phylogeny tree, you're trying to track the progression of the tumor as a whole. And so what changes occurred first in the tumor, and then what changes occurred later in the tumor's progression. And so when we get this DNA sequencing, you can sort of look at the frequency of changes for a particular mutation. And so what we use is we compare those. So if you have two genes, say IDH1 and TP53, that have a similar frequency of mutations across the genome, across all the cells, you can assume that they occurred around the same time, or that's what the theory of this is. Whereas if a certain mutation has a lower frequency, you can assume it happened afterwards. And so what you're trying to identify is the mutations that occurred in 100% of the cells, and we call those clonal. And so we're defining different tumor clones by the fraction of mutations that have similar numbers. So like, let's say these, what we're defining in blue here, these are important mutations to the genome, sorry about that, we're estimating them as having occurred in all the cells. And so these what can be defined as clonal. These mutations occurred first, and these genetic changes led to a more productive scenario for other mutations to occur and for the cell to survive despite whatever environmental conditions or treatment may occur. And so you have certain genes that you see across different samples, across different patients, that we see occurring first. And so those are what we call driver mutations, because they keep showing up as the first ones that are changing with this particular type of tumor. And
Yeah, so they're like driving the tumor genesis, driving the cancer progression. So I guess if I could say that back a little bit, like we're looking at that gray cell at the top that has 0 mutations. And if we follow this arrow down in the progression of that cell over time, maybe it accumulates some of these mutations that drive it. These are the driving mutations that make it, whatever, divide faster, not have as many cell checks, and that causes it to be a tumor. And then that's the progression that you're measuring.
Yes. So looking from the top to the bottom, you'll see you start out at 0 mutations, and then you see this accumulation of mutations over time where certain key ones occur. TP53, for instance, this occurs in a lot of tumors, and this particular gene is part of the error checking process when your cells are replicating. And so normally before your cells divide, there's a point where the cell sort of stops and evaluates, is it healthy, and are these, when the divisions occur, are these divisions properly? Is a C being paired with a T? And if not, can we fix that base pair, or say there's DNA damage, can we repair the damage? And so one of the first mutations you see is in this error checking gene, where as opposed to stopping to repair, damaged DNA or mismatches in the genetic code, this gene being mutated, your cell is less likely to do that. And so it'll just keep pushing through errors and then allowing other mutations to occur, which can be more advantageous to the tumor cell.
Yeah, I see. So that's how it progresses into this bad tumor.
Right. And so what we're looking at, like for instance, the way, I don't know if our bubble here is covering it, but in the bottom right, we have the fraction of tumor cells of the particular set of mutations. And so over here, these mutations, so some 4,000 are affecting all of the cells. And then you see certain subsets getting specific other mutations that are important to the development of the tumor. And so when you're tracking, say, which treatments you want, if you want to target a specific gene, it's important to know how many of these cells actually have that gene that you're targeting with a particular drug. And so
This
this
is
is
similar to what we saw on the previous slide, where you had, even within one tumor, you kind of identify these different classes, these different categories. And so that's kind of what you're seeing now with the mutations too, that a lot of them will have the same mutations, but some will have other ones. So these are your different clonal populations that you're identifying.
right, yes. And so we can use these tools to sort of identify how this tumor is progressing over time and what changes are occurring and how it's affecting the molecular background of the tumor.
Yeah, and this is all from genetics. So from sequencing what the DNA looks like and what mutations you identify across all these different tumor samples.
Mhm.
Cool, very cool.
Thanks.
So what does a typical day look like for your job? It sounds like it might be very different day to day because you do all these different types of techniques and you have different skills that you incorporate.
Yeah, so I'm working on a few different projects at any given time. And so the day changes depending on what I'm working on. It's not a very rigid schedule, which I kind of like actually. Um, so on a given day, depending on what project I'm working on, I spend a lot of time writing software code or to analyze the data. So like either running different workflows to get the sequencing data to the point where we can do these analyses, or just running different experiments through writing and adapting different software programs to sort of analyze these patterns. And then I'll spend some time reading papers, I'll have meetings with people. And so like we might need to sit down and plan future experiments, talk about stuff that's happened before. Sometimes it's just with my boss, sometimes it's just with people I'm working with, sometimes it's both. And then also when the project finishes, you have to spend time writing, and there's a lot of writing and reading to be done. And so it changes up a good bit. It's nice.
Yeah, and a lot of collaboration, it sounds like, between your scientific colleagues, a lot of solo time to do your own research.
Mhm.
It's very,
Yes.
very different every day.
And it's, it's in an office setting, but it's not a firm 9 to 5. I may have a meeting earlier this day, and so I may start sooner, or I may be caught up in something, so I may work later. And so the timing's not necessarily rigid like an office setting, but I am working in an office. And so, like here, I can come into my desk and maybe I'll run into somebody and it can turn into something. Like you have to sit down and like, oh, go over this, or we have Slack. And so a lot of times, like for instance, earlier today, so one of my friends hit me up about something we're working on. And so we'll just sit and have a quick meeting to sort of talk about results and stuff. So it changes a lot day to day.
Yeah, and I think sometimes like having those little interactions, like about anything or even research, can help spark new ideas, right? Like you're saying, oh yeah, I'm working on this project, and maybe your friend or your colleague might have an idea as you're explaining it, or you can chit chat about it, and
Yeah.
you can find some really, really good progress, like get some really good progress that way.
And then another big thing, just on that idea of getting ideas, is that you often, so like for instance, at Jack's, people host talks, or at like you might look at, like there's different, especially in this current pandemic situation, there's a lot of online talks. And so you can go attend someone else's talk, or maybe there's a webinar about some analysis technique. And so there's a lot of chances to talk to other people in the general field, or just get ideas or hear what's new that's happening in the field. And so there's also a good bit of that happening as well.
Yeah, absolutely. Hear a talk, watch a seminar or something like that, and get inspired.
Yeah.
Yeah, along those lines, do you have any kind of parting words of wisdom for anyone looking to start a career in cancer research or in computational research or any sort of STEM field?
I think one of the things that I spent a lot of time being anxious over, sort of coming up, was like knowing what I wanted to do. Like, what's that big? Like, what do you want to be? What do you want to research?
Yeah.
And I would just say that you can break it down into smaller chunks so that the whole question's not so overwhelming. You don't have to know exactly what you're going to do, but you can narrow down. What do you like? What don't you like? What areas do you have familiarity with? What do you feel the most confident in? And then you can move in that direction, and it's okay to change it. I think some people may feel like, oh, I've, spent this time in this lab, I should do this thing now. And, you have time to adjust it as long as you've learned something from it. it can be a valuable experience, even if you're not ever going to work on that thing again. I've worked in several topics for things that, like, I'm probably never doing molecular dynamics again. And that's okay. I learned some valuable things. I can have some messages I can take away from it, and then I can, find a way to use it later. So
Yeah,
it's,
absolutely. That's really good advice too. And it kind of takes some pressure off that you don't have to plan everything, know exactly what you want to do, but just take like one step at a time.
yeah, yeah. And people around you can be helpful. You can add, professors may know what your talents are. You may have advisors that should give you class ideas, and maybe your friends notice that you like doing something more. Like maybe you come home and you complain about something in particular, or maybe you'll show your friends something in particular, and that can be a hint.
Yeah, absolutely. And sometimes too, like knowing what you don't like is almost as important as knowing what you do like or what you aren't good at or something. So that can also be a lot of help.
Definitely.
So kind of along those lines, what's next for you? Where do you see your future? What do you want to do when you grow up?
Oh man, I've still been that's what all the school's for, so I can push off growing up.
Exactly.
I think I'm working more towards working on research in more of an industry setting, to be honest. And again, I guess this is sort of my penchant for trying out things. I have been more or less accustomed to the academic life, and I'm sort of curious what a more direct workflow is in people within the field where it's more tangible timelines. And I've been working in what you would call basic science, where I'm sort of answering broader questions about overall mechanisms, which is pretty important. But I'm curious what it would be like to work where the questions are more direct and translatable to the patient's in condition. And so, yeah, so basically I came to Jax to try to build more broadly applicable skills. What I worked on in grad school was it was a very niche technique for a specific research question, which was cool, but I'm trying to branch out a little bit.
Yeah, so the comparison would be like industry would have more, like you said, direct kind of contacts or timelines to patient clinical like outcomes. And it sounds like your work now, at least with some collaborations with the clinicians that are giving you tumor biopsies and you processing patient samples, is a good skill set for you, like to help get you there.
Oh yeah, yeah. I've tried to make it. I guess I've more or less tried to keep doors open as much as possible. And Jax has been a great place for that.
Great. Do you have any parting words before we close this chat?
Um,
You gave some good words of advice, so no pressure to say more.
hmm. I'm trying to think. I think one thing is that when you work on this sort of thing, you're gonna come across. I mean, almost nothing is gonna go smoothly, to be honest, and that's something to just be prepared for. It's pretty much expected. You're gonna have an idea. It should work out on paper, or maybe there's theory behind it, and stuff's gonna pop up. And I guess that's where the meat of the problem solving comes in, is how you deal with the unexpected of it, and managing that. It can feel frustrating sometimes if you don't get the results you're looking for, and sometimes it can be months, sometimes it can be longer. But I guess sort of knowing that coming in, it can take a long time to get some of this stuff right. But you keep working at it, you stay dedicated, you talk to people, and you ask for help, and you'll get there. But yeah, so I guess just sort of getting expected to a lot of missteps and what you might consider failures. You just kind of have to think about it as it's roadblocks for you moving forward.
That's really good advice and very inspiring. And thanks so much for being here, Kevin, and sharing your research. I learned so much. I loved seeing your figures, and it was really fascinating to see just how different within one tumor the cells can be and all the techniques that you're using in order to analyze that. And it makes me really hopeful too for therapeutics in the future. It really just is a game changer, it sounds like.
Yeah. Well, thanks for having me.
Yeah, and thanks for sharing your career journey and all the cool stuff that you're doing. And to our viewers, thanks so much for tuning into this episode. Don't forget to watch more interviews from the Career Chat series. Until next time, bye-bye.